Hi, welcome to the GPAT preparation series 2 test number 4. This test series will be helpful in preparation for the GPAT exam. We wish you all the best. Before we start the test, let us understand the process. So, here is the process. This test consists of 10 questions and you will get 7 seconds to write your answer in the comments. For example, if the answer to question 1 is option B, you can write in the comments as 1B. Similarly, if the answer to question 2 is option A, you can write in the comments as 2A. So, the first question is, a Chinese tree Camptotheca acuminate is useful in cancer chemotherapy. The drug selected above acts by option inhibiting topoisomerase I, option B inhibiting topoisomerase II, option C inhibiting thymidylate synthase, option D forming hydrogen peroxide which generates free radicals. As you know, you have got 7 seconds. If you know the answer, mention it in the comments. The time starts now. Time is over. I hope you got it right. The correct option is that is inhibiting topoisomerase I. Now let us quickly move to question number 2. They can interact by different mechanisms. Option A, biological fluid diffuses into the matrix and causes erosion of polymer. Option B, endocytosis by phagocytic cells of the reticuloendothelial system such as macrophages and neutrophils. Option C, magnetic beads disperse throughout the polymer matrix. On exposure, the drug is released slowly by diffusion. Option D, receptor binding mediated by the peptide. You have got 7 seconds. If you know the answer, mention it in the comments. The time starts now. So, the time is over. Let us see the correct answer. The right option is B, which is endocytosis by phagocytic cells of the reticuloendothelial system such as macrophages and neutrophils. I hope you are participating in the test and writing the answers in the comments. That is awesome. Let us move ahead. So, the next question is, liposomes are used as carriers for drugs and macromolecules in pharmaceutical formulations. They are option A, phospholipids dispersed gently in aqueous medium to obtain multilamellar vesicles. Option B, hydrophilic or lipophilic polymer matrix with a drug reservoir. Option C, a shallow compartment molded from a drug impermeable system and rate-controlling polymeric membrane. Option D, microporous membrane made from ethylene vinyl acetate polymer. Mention your answer in the comments. You have got 7 seconds. Let us see the correct answer. The right option is that is phospholipids dispersed gently in aqueous medium to obtain multilamellar vesicles. I hope you are ready to take the next question. Here it is. Transgenic plants are developed by genetic engineering techniques. Captopril IP is assayed by titration. Option against 0.1 N sodium hydroxide using phenolphthalein indicator. Option B, of a solution in diethylformamide with 0.1 M of tetrabutyl ammonium hydroxide. Option C, of a solution in anhydrous formic acid and acetic anhydride with 0.1 N perchloric acid. Option D, of a solution containing 1,8N sulfuric acid and potassium iodide with 0.25 N potassium iodate using starch solution. Mention your answer in the comments. Your time starts now. Let us see the right answer. The right option is C, that is of a solution containing 1,8N sulfuric acid and potassium iodide with 0.25 N potassium iodate using starch solution. Here is the next question. Let us see if you know the answer. Transgenic plants are developed by genetic engineering techniques in the design of Captopril. The option A, COOH group is introduced in proline to enhance the binding capability at the receptor site. Option B, SH group is introduced to enhance the binding capability of the drug with cobalt ionovase. Option C, SH group is introduced to enhance the binding to the zinc ionovase. Option D, COOH and SH groups to introduce hydrophilic pockets at the receptor site. Your time starts now. Here is the answer. The right option is C, that is SH group is introduced to enhance the binding to the zinc ionovase. Few more questions left. Let's move ahead and take the challenge. Transgenic plants are developed by genetic engineering techniques in the production of transgenic plants. The gene transfer is carried out by option induction of meristematic primordial, option B, gel filtration, option C, clonal propagation, option D, silicon carbide whiskers. Your time starts now. The right option is D, that is silicon carbide whiskers. Few more questions left. Let's move ahead and take the challenge. Transgenic plants are developed by genetic engineering techniques. The method involves option individual genes from one species inserted into another. The offspring will contain copies of new gene. Option B, by crossing two species or varieties differing at least in one set of characters. Option C, exposing the plant tissue to radiation. Option D, bioproduction of natural compounds under aseptic conditions. Only 7 seconds left. Post your answer in comments. The right option is that is individual genes from one species inserted into another. The offspring will contain copies of new gene. Wow, you are participating really well in the test. Let us see if you can answer the next one. Group I, Group 2. Condition description. P. Agranulocytosis 1. Reduced lifespan of erythrocytes. Q. Anisocytosis 2. Lack of neutrophils. R. Plastic anemia 3. Abnormal variation in RBC size. S. Hemolytic anemia 4. Depression of synthesis or cell types in bone marrow. Option A P2, Q3, R4, S1. Option B P2, Q4, R3, S1. Option C P1, Q2, R4, S3. Option D P4, Q2, R1, S3. Your time starts now. I hope you could find the right one. Only 7 seconds left. Post your answer in comments. The right option is that is P2, Q3, R4, S1. Here is the next one. Group I, Group 2. Microorganism typical characteristics. P. Coinbacterium diphtheriae 1. Cells divide in 3 planes in an irregular pattern, producing bunches. Q. Streptococcus pyogenes 2. Cells are lined side by side like matchsticks and angles to one another. R. Staphylococcus aureus 3. Long, branched multinuclear filaments called hyphae. S. Streptomyces viridochroma 4. Cells divide in one plane and remain attached to form chain. Option A P3, Q4, R2, S1. Option B P4, Q1, R2, S3. Option C P2, Q4, R1, S3. Option D P3, Q2, R1, S4. Your time starts now. I hope you could find the right one. Only 7 seconds left. Post your answer in comments. The right option is C, that is P2, Q4, R1, S3. Here is the last one. Give it a try. Group I, Group 2. Process required molecules. P. Post-translation modification 1. Signal peptidase. Q. DNA repair 2. Signal factor. R. Control of prokaryotic transcription 3. Proteasome complex. S. Protein degradation 4. Photolyase. Option of P1, Q4, R2, S3. Option B P2, Q3, R1, S4. Option C P3, Q2, R4, S1. Option D P2, Q1, R3, S4. Your time starts now. I hope you could find the right one. Only 7 seconds left. Post your answer in comments. The right option is that is P1, Q4, R2, S3. Since you participated in all the questions of this GPAT preparation series 2 test number 4, we really appreciate your effort. We wish you all the best for your GPAT preparation. See you soon in the next GPAT preparation series video.